Hey, welcome back to Bio 100. We're going to do another lecture today. We're going to continue on with a little bit more about cancer and then meiosis. So last time we left off, we were talking about the different phases. We have interphase we can start with G0, G1, S phase, and G2, and then mitosis and cytokinesis. And this is kind of the layout and proportionally how often a cell or how much time a cell spends in each one of the phases. And what I want to do is just start off class today. I want you to hit the pause button and take a second and see if you can identify the different stages of mitosis and even cytokinesis in this particular image. Okay, hopefully you took the time to find those different stages. If you really struggled, I want you to just take a second and focus in on this and teach yourself. There's even some links that you can take here, looking at prophase, metaphase, anaphase, and telophase and cytokinesis. And then let's revisit this. So go back, hit the pause button, watch the link, do whatever you need to do, and then come back and see if you can identify them here. What's going on here? What's going on here? You've got some stuff pretty far along here. You can see the origins. This is in a plant. Keep that in mind. See the origins of a cell wall there. You should be able to kind of go through and be able to find some good examples of each one of those. Okay. So mitosis gone bad. We've talked about how this results in cancer, and so what we should probably hit on is how your body can stop it, right? Why don't all of us have cancer? We know that a huge chunk of us are going to get cancer, supposedly throughout our lifetime. About 20 to 25% of us will pass away from cancer. So what does our body do to stop it so that all of us don't eventually die from this? Well, luckily, cellular division is a really tightly controlled process. Normal cells have the ability to actually halt cellular division at checkpoints, and these are essentially places like checkpoints throughout the cell cycle where proteins can survey the conditions of a cell, and the cell has to pass the survey or it will be destroyed or not continue through cellular division. So let's go over each one of these. So you have what we call the G1 checkpoint. Remember, you come out of G0 into G1, so there's the G1 checkpoint, and it's really just looking for damaged DNA. The cell it's also got to have a large enough, it's also got to be large enough and have a good amount of nutrients to pass through cellular division. So the G2 checkpoint needs to make sure it checks that the DNA is properly replicated. And then last but not least, the metaphase checkpoint have all the chromosomes attached properly to microtubules so that they can be pulled to the ends of the cell properly into sister cells. So the problem is that you have a set of genes that can throw this off, and these are called oncogenes. Have you ever heard an oncologist, somebody who studies cancer? So these would be proto-cancer genes, right? So these are genes that are going to produce proteins to control the cell cycle. And then you have mutations. So this is a change in the sequence of DNA. So mutations can be inherited or caused by carcinogens. This changes the structure and function of the proteins. So mutated proto-oncogenes become essentially just oncogenes. They no longer have the ability to regulate cellular division. So they usually, not only do they not regulate it, they usually overstimulate cellular division. So here's your regularly healthy looking proto-oncogene. Everything's great, DNA is replicating wonderfully. Then you get this mutation, and you see, like in this example, right, this valley that's present in this protein is no longer here. And this may lead to overstimulation of cellular division as it's overriding the checkpoints. So there are also tumor suppressor genes. These are genes that stop cell division if conditions are not favorable. They can allow, but just like with the oncogenes, when the tumor suppressor genes are mutated, they can actually override checkpoints as well. So again, here's your healthy tumor suppressor protein. checking out the DNA, everything's good, but then you get a mutation here, and this it fails to stop any sort of growth because these two proteins no longer look the same, they can't regulate the same, their composition has been changed. So you really need a lot of mutations for cancer to develop. It's not just one, it's much more than, it's usually more than that. And so you have these benign tumors that will essentially require many mutations for them to become cancerous. So here's some steps. Angiogenesis, the tumor gets its own blood supply. It's never a good thing. The loss of contact inhibition, cells will now pile up on each other, whereas normal cells won't usually pile up on each other, or they'll pile up on each other in a controlled manner, but that doesn't happen in tumors. They can continue to pile up. Not only do they pile up on each other, but there's this loss of anchor dependence. So normal cells have a controlled way that they pile on top of each other, and they're all independent, they're all dependent on each other, they're anchored together. But when you lose that anchor dependence, it allows these cancer cells to essentially move around the body and establish new tumors. And then cells become immortalized, and that there's no fixed number of cellular divisions for each cell. And here's a fantastic video that we'll watch here.
There is a huge fundraising deadline coming up
I apologize.
for Senate Democrats, and they need your help right now.
Cancer is a group of over 100 diseases characterized by abnormal, uncontrolled cell growth. In a healthy body, cells grow, die, and are replaced in a very controlled way. Damage or change in the genetic material of cells by environmental or internal factors sometimes results in cells that do not die and continue to multiply until a mass of cancer cells or a tumor develops.
You can see the blood supply coming
Most
in there, right?
cancer-related deaths are due to
Cells
metastasis,
pile up,
malignant cells that penetrate
one
into the
goes away.
circulatory system and establish colonies in other parts of the body. Great advancements have been made, but cancer is still the leading cause of death for people under the age of 85.
So, stay healthy, exercise, eat well, and things will go better for you. So another thing to think about here is that not only does it require a lot for cancer to pass throughout the body, but it requires a lot for cancer to develop, to even start to develop. So there's this idea of this multiple hit model, and that each mutation within your DNAs can be considered as a hit. And each added mutation or hit is what creates the tumor potential. So some mutations can be inherited via gametes, and those can cause cancer, although they're very rare. This is something you consider familial risk. What's more likely is that you inherit the possibility of having a mutation. Most hits are probably acquired during a person's lifetime, so not familial risk. So meaning that this is more likely you're exposed to things, you're not exercising, you're not eating well, you're not taking care of yourself. These are most likely the reasons that contribute to cancer. So with that, we'll leave the cancer. We'll move on to meiosis a bit. How is meiosis different than mitosis? I want you to just take a second, think about that, see if you can figure out in your mind how meiosis and mitosis are different. Okay, so meiosis is essentially a specialized form of cell division. It happens in the gonads to produce gametes. It's going to reduce the number of chromosomes in each cell by 1/2, and then chromosomes come in homologous pairs, as we've talked about. So gametes get one pair of each. So gametes are haploid, meaning that because they only have one of either the parent or the mother chromosome, that they're going to be haploid, which just means N, and then somatic cells, which are all the other cells in your body, are going to have both copies, one from mom, one from dad, so they're going to be diploid. So before meiosis, there's an interphase step. The DNA is duplicated. Meiosis takes place in 2 stages after this, meiosis 1 and meiosis 2. So in meiosis 1, it's going to separate the duplicated chromosomes, one from dad and one from mom, right? It's going to separate them from each other into 2 cells. And then during prophase 1, there may be crossing over between chromosomes of homologous pairs, and after meiosis 1, the resulting cells are going to be haploid, and they're going to continue on into meiosis 2, which is going to separate the sister chromatids of a homologous pair into individual cells. One single chromosome, so one single sister chromatid per cell, and it's resulting in sex cells, either sperm or egg in humans. So here's kind of what we're looking at, right? So we have this end, right? There's been duplicated DNA, end of interphase, and they enter into prophase 1, okay? And it's just like prophase, metaphase, anaphase, just like in mitosis, if you think about it. Now the one difference is that here in prophase, there's something that can happen called crossing over. And we'll talk a little bit more about what that is in a minute, but essentially it is the same length of these chromosomes can be passed off between each other, right? And you can see that it's happened here. There's been crossing over here where these chromosomes have swapped out portions, equal portions between each other. Okay, so it's really important to look, where does crossing over take place? Okay, and then in meiosis 2, you have prophase, metaphase, anaphase, and then telophase and cytokinesis. Okay, and you can kind of see how here at the end of meiosis 1, you have cytokinesis. At the end of meiosis 2, you have cytokinesis. So I want you to take a look at this, hit the pause button, soak this up, go back to the other slide if you need to, and just make sure you have a good feel of what's happening here. Okay, hopefully you're back. I want you to look at 6. Is there any amount of crossing over happening there in step 6 or prophase 2? All right, so here's a cool video that we're going to watch, and hopefully this will help straighten things out, hear it a little bit differently from somebody else's take.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The 2 homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in 2. This second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
All right, so let's keep moving on. So let's do an activity. I'd like you to take out just a piece of paper. Okay, so go ahead and hit the pause button, find a piece of paper. Okay, so here are your instructions. I want you to draw mitosis on the top left. So take that piece of paper, fold it into 4. And on the top left, I want you to draw out mitosis. On the bottom left, draw out meiosis. And then on the bottom right, draw out meiosis 2. And this should give you, well, we'll talk about what it'll give you in a second, but I want you to answer these questions in the remaining open space, which should be your top right. How many mitosis, so how are mitosis and meiosis similar? How are they different? What is crossing over, and at what point in the cycle does it happen? Go ahead and hit the pause button. Take the time and draw this out. You're going to want to do this. Okay, hopefully you're done with that and you've taken it seriously and knocked it out. So let's talk about crossing over. So we've mentioned it a little bit. I've given you kind of a cursory description of it. So crossing over is really the exchange of equivalent portions of chromosomes between members of homologous pairs. So when does it happen? You should just know this by now. I'll let you answer that if you don't. So it results in new types of gametes being formed. So here is an example. Let's start with the no crossing over, right? You go through metaphase 1, metaphase 2, daughter cells. And essentially what you're looking at here, there's been no crossing over, but you get 2 genetically identical cells as a result of what happens without any crossing over. But with crossing over, you can have these chromosomes exchange equal portions with each other. So you can see this is the result right here, okay? And then as they continue through meiosis, those daughter cells are going to be completely distinct. So you end up with 4 distinct daughter cells in this example, whereas over here you only had 2 sets of distinct cells. Also, there's a random alignment. So you have these chromosomes from mom and these chromosomes from dad, but when they get pushed to the middle for alignment, it doesn't matter which side they align. The only thing that matters is that the chromosomes move to the middle. So it can result in different types of gametes just by the way they align. So here's an example of them aligning perfectly with the paternal chromosomes on one side, the maternal chromosomes on the other. As they move through meiosis, you come down here and you have just 2 different combinations. Okay, so this is combination 1, combination 2. But if they align randomly without any crossing over whatsoever, they just align randomly, you can see that you can get a 3rd and 4th combination. Okay, so here are some suggestions, lots of links for you guys to go in, look at these. Some of these we've seen, some of them you haven't, but hopefully they're helpful to you in your understanding of meiosis, crossing over, random alignment. And then there can be some mistakes in meiosis. So we've talked about in mitosis, there can be mistakes. There can be also mutations in the genes that regulate cellular division, and these can all lead to cancer. But in meiosis, we have this thing called non-disjunction. This is really just the failure of homolog chromosomes to separate normally during meiosis. And this results in gametes that have too many or too few chromosomes. So you've probably heard of trisomy 21. A huge chunk of you probably know exactly what that is. There's also monosomy. Most embryos that result from these gametes will die result from the union of these non-disjunct gametes. They'll die before birth. But there are a few chromosome abnormalities that can make it to life. It happens all the time in plants and sometimes in humans. And here are some examples. So in plants, when you're looking at these, this is the wild type, this is the diploid individual, and then these are the trisomic individuals here. And you'll notice one of these poinsettias you see a lot of around Christmas time. And then in humans, this is a monosomy example. I believe this is monosomy 16. I'm not entirely sure, although I think that one's fairly lethal. In any case, you have a monosomic example here, and you'll see that these individuals, although they look healthy and operative, you can just tell by looking at them, there's just something that's a little bit lacking there in terms of the way their faces are structured. There's also mental disabilities as well, generally. And then trisomy 21, Here, this is Down syndrome, which some of you may have Down syndromes in your home. Us Down syndrome individuals, that you know, brothers and sisters, the uncles and aunts that live around you, and they're a total blast. I've had some fantastic interactions with Down syndrome individuals, but those individuals just have a third copy of a chromosome at the 21st chromosome. There's trisomy 18 here and trisomy 13 here. So with that, we'll kind of leave with this, because I know I've mentioned this already, but students always have these burning questions. Can cancer mutations be passed on to offspring? So mutations must be present in the cells that give rise to gametes if they're going to be passed on to the offspring. Mutation in somatic cells like skin cells are not heritable, although the predisposition for skin cancer might be heritable. You can't inherit the mutations that happen in skin, that happen in the skin of an individual. Okay, and then here we'll just leave off. Here's some cool pictures of sperm and egg coming together, the possibility of genetics here, and the possibility of two separate sets of genetics coming together, and all of the possibilities that provides. And next time we'll start getting into the genetics. I'm pretty excited, and I hope that this was helpful to you. If you have any questions, email your TAs. They've been very helpful on top of things, and I hope you have a fantastic day.